In this video, we will explore the F420 program pipeline at Ayass Research Institute. The F420 software takes a CSV file as input and in a matter of seconds produces a customized report. However, the complex system running in the background encompasses many steps: normalization, unsupervised learning with PCA, model fitting, functional analysis, machine learning for multi-disease diagnosis, machine learning diagnostics, patient classification, and creating personalized report. The median of ratios method provides an intuitive and straightforward approach to normalizing RNA-seq count data. Let me walk through how this normalization procedure works. First, we calculate ratio of each gene's count to the mean count for that gene across all samples. This gives us a set of gene-wise ratios per sample, representing the fold change of each gene relative to its average expression. Next, we take the logarithm, typically log base 2 of each gene's ratio for every sample. Logging the ratios makes them symmetric around 0 and normally distributed. Now we have a matrix of logged fold changes for each gene in each sample compared to the mean. To summarize this into a single normalization factor per sample, we calculate the median of the logged ratios for each sample. The sample-wise medians encapsulate the overall shift of each sample relative to the mean. We use the median because it is robust to genes with extreme high or low ratios. Finally, to normalize the data, we divide each gene's raw count by the median of ratio's normalization factor for that sample. This centers the distributions while preserving relative differences between samples. Let me describe how we could apply principal component analysis, or PCA, to reduce dimensions and visualize relationships in our transcriptomic data. First, we input our normalized expression matrix, with genes as the rows and samples as the columns. Next, we standardize the data so that everything has mean 0 and variance 1. Then, we calculate the covariance matrix containing the covariance between every pair of genes. Decomposing this covariance matrix gives us the principal component axes, ranked by the amount of variance they explain. By projecting each sample onto the first few principal component axes, we can visualize them in a reduced PCA space. Samples close together have more similar expression profiles. We can also examine the gene loadings on the top PCs to reveal clusters of correlated genes driving phenotypic differences. Overall, PCA provides an unbiased, data-driven overview of the 18,000-plus gene transcriptomic landscape. It highlights the major sources of variation and how samples relate in high-dimensional expression space. When analyzing differential expression in RNA sequencing data, we need to fit an appropriate statistical model to the count data. Next, we model the count data for each gene across samples using a negative binomial distribution. The negative binomial captures high variability better than a simpler Poisson model. We incorporate the sample-specific size factors into the mean of the negative binomial model as an offset. This models the mean expression while accounting for library size. The negative binomial uses a dispersion parameter to model variance as a function of the mean. We estimate gene-specific dispersion parameters to allow for over-dispersion. Finally, we fit the full model including size factors and dispersions using maximum likelihood estimation. This gives normalized mean expression estimates for each gene in each sample. The negative binomial modeled counts reflect true biological variability instead of noise from library size differences. Downstream differential expression analysis is then performed using the fitted mean expression values and estimated dispersions. Functional analysis is crucial for interpreting lists of differentially expressed genes from transcriptomic studies in a biological context. We start by identifying genes that are significantly up or down regulated between conditions of interest. Then we input these gene lists into bioinformatics software or ingenuity pathway analysis. These leverage databases of pathways, gene ontologies, protein-protein interactions, hub gene analysis, and more to identify functional enrichments. We get back results ranked by statistical significance. By piecing together these enriched functions and pathways, we can start to understand the coordinated molecular systems that are altered in patients. Overall, integrating functional analysis with differential expression profiling focuses our analysis on mechanistic insights instead of just lists of genes. Machine learning provides a powerful data-driven approach for classifying multiple diseases from transcriptomic data. First, we took the already processed patient data with transcriptomic profiles and known diagnoses to train the models. Then we trained various classifiers like logistic regression, SVMs, random forests, and neural networks on the training data. Ensemble methods that combine multiple basic models can improve robustness. We optimize hyperparameters like regularization and tree depth through cross-validation. Once the models are tuned, we evaluate classifier performance on the test set using metrics like accuracy, F420 scores, and AUC. To gain insight into the models, we can analyze feature importance, but it's important to note that we have carefully selected these features based on genes that best fit the disease classification. Multi-disease classification is inherently challenging due to heterogeneity within and similarities between diseases. But large data sets and model ensembling can improve accuracy and enhance the already established bioinformatic work. Most importantly, rigorous validation on held-out patients prevents overfitting and ensures generalizability. Overall, our extra layer of machine learning provides a powerful data-driven approach to leveraging transcriptomics. Thoroughly evaluating the accuracy of machine learning classifiers for transcriptomic disease diagnosis involves multiple analyses. First, we report overall performance metrics on the test set, like accuracy, F420 score, and AUC, noting performance relative to baseline models. Looking at the confusion matrix can reveal which classes are commonly misclassified and why. And we can calculate diagnostic performance metrics like precision, recall, specificity, and sensitivity for each individual class. Beyond this, generating ROC and precision recall curves by thresholding the model probabilities provides a more complete picture of performance over all thresholds. To identify biases, we can break down performance by patient subgroups, analyze frequently misclassified cases, and audit prediction errors. And model calibration techniques assess how well-aligned predicted and true diagnosis probabilities are. No model is perfect, so thoroughly investigating biases and trade-offs is crucial for clinical acceptability. Continuous monitoring of diagnostic performance post-deployment detects potential concept drift as new data accrues. Overall, rigorous validation from multiple angles provides confidence in model accuracy and generalizability. The goal is to provide an auto-generated personalized report for each patient that extracts biological insights from their transcriptomic profile. First, we classify the patient's transcriptomic data using tuned machine learning models to better characterize their disease and potential subtype. This goes beyond traditional diagnosis to reveal new stratifications. Next, we perform pathway and gene set enrichment analysis on their differentially expressed genes to identify key biological processes dysregulated. This reveals the underlying molecular mechanisms at play. Then we use knowledge bases and network analysis to pinpoint possible drug targets along those pathways. Finally, we cross-reference with databases of approved drugs to match targeted therapies or clinical trials. The personalized report highlights the patient's disease classification, dysfunctional pathways, and potential precision medicine interventions tailored to their specific transcriptomic profile. It transforms complex omic data into clear, actionable insights for clinicians and patients. The focus is on providing additional clarity and answers not found through standard practices. Overall, combining cutting-edge bioinformatics and machine learning with interpretable reporting enables truly personalized systems medicine. That concludes our presentation. Thank you for your interest in our program. For more information, please visit our website f420.ai or call 888-889-4770. Stay safe, and we look forward to seeing you in future videos.